Our next speaker is Dr. Vilhelm Bohr from the University of Copenhagen. Dr. Bohr is one of the world's most published researchers on aging and neurodegenerative diseases with over 600 publications. For the last 30 years, he served as the chief of the Laboratory of Molecular Genetics at NIA. He has now retired and now works for University of Copenhagen, but he still lives in Maryland. Dr. Bohr earned his MD, PhD, and DSc degrees at the University of Copenhagen. Throughout his career, Dr. Bohr has made significant contributions and advanced understanding of DNA repair pathways, mechanisms, and cellular response to oxidative DNA damage and oxidative stress. A decade ago, Dr. Bohr initiated research through the industry-leading Chromadex external research program evaluating Iogen, their patented nicotinamide riboside chloride. His research has been conducted in models of Alzheimer's disease, age-related hearing loss, and other conditions resulting from age-related decline. Today, he is going to share his work on hearing loss in Alzheimer's mouse models.
Thank you so much. Really a pleasure to be here in person. I have spoke remotely at one of these meetings. It's really exciting. Thank you for having me. So I will try to talk a little about some different things that relate to hearing loss, including DNA damage and repair, and then NAD substitution, and then mitochondrial function and Alzheimer's disease. Firstly, I would like to review the DNA damage response because we have a tremendous amount of damage to DNA, and there's a lot of thinking right now that this is really the driver of aging. Hundreds of thousands of lesions per DNA, per cell, per day, and that causes a lot of detriments unless they're very efficiently repaired. So if we have a DNA damage or a change, an alteration in DNA that is seen as a DNA damage, and there are many others than just regular known lesions, it can be repaired by different repair pathways. I'll talk a little about that, but it can also be handled by a variety of other systems. Importantly, DNA damage responses involve a lot of cellular signaling, including chromatin structural pathways, inflammation, and many more. And the endpoint of lack of these things is senescence, aging, neurodegeneration, and cancer. So this DNA damage is very profound. We can have changes in transcription, changes in replication stress, and then there's another signaling pathway which in our lab we have been concentrating on the last several years, and that is a pathway through mitochondrial dysfunction that I'll discuss a little more. So that the idea is that DNA damage causes signaling pyrrolation that then causes mitochondrial dysfunction, and this leads to mitophagy or other deficits in mitochondrial function that ultimately lead to these endpoints as well. One of the model systems that some model systems that we have utilized a lot in the work that we have done are these segmental progeria, premature aging diseases, where the patients appear much older than they actually are. And these are very interesting model systems that have been widely used to study aging. One patient type is Werner syndrome, which is quite well known for its mimicking of the aging process at so many levels. The patients appear much older than they actually are. Cockayne syndrome also, I'll talk a little more about that. There's a skin disease, xeroderma pigmentosum, and then there's Rothman-Thompson's disease. These are, together with Werner syndrome, RECQ helicase deficiency, so deficiency in RECQ function. And then Hutchinson-Gilford and ataxia. These are all premature aging conditions, and they all have been used widely for model systems to understand mechanism and also the aging process. And importantly, they all have deficits in DNA damage processing, DNA repair processes. So it also supports the notion that if you have a deficiency in DNA damage processing, that can lead to progressive aging. And so we studied a number of these diseases that are particularly prone to get neurodegeneration, and found in a large number of papers that they all had in common mitochondrial dysfunction And here's a mitochondrion without mitophagy, lacking mitophagy, so damaged mitochondria are not removed by mitophagy, remain in the system. And this signaling that we found in mitochondria were things like increased pyrrolation, which I'll talk a little more about, a DNA damage signaling process, and mitochondrial ROS formation, membrane potential changes, this mitophagy problem, and then lower levels of NAD. And these then led to sirtuin activities were decreased, and many clinical features in these diseases are similar to what you see in other known mitochondrial diseases. And later on, we also characterized Werner syndrome, apotaxia, and more lately found many of these features in Alzheimer's disease using model systems. So the assumption is that when you have DNA damage, here's a double-strand break in DNA, you have this post-translational modification called pyrrolation. This occurs in the genome, various sites in the cell, and in mitochondria, which we have worked with recently. And this depletes NAD pools because it uses NAD as a substrate, and you get a depletion of NAD leading to other enzymes that need NAD, and many enzymes do. It's an essential substrate that then causes various dysfunctions, including the mitochondrial dysfunction. NAD is, as I mentioned, an essential substrate, and it is part of the metabolism in the mitochondria and in the nucleus. And it also underlies deficiencies in NAD have been shown to underlie many of the so-called hallmarks of aging. These are the hallmarks of aging that were described fairly recently as a measure of aging underlying mechanisms. So DNA damage, processing, telomere attrition, various of these things, and actually all of them at this point are somewhat related to NAD pools and NAD function. And it's been shown in mouse models and in part in humans that our pools of NAD decline with aging substantially. So this has led us to a model that we've worked with in different of these diseases and more general in aging. You have DNA repair decline, which has been shown to take place in aging, about 1% per year in humans. And then in these particular diseases of lack of DNA repair, and Alzheimer's disease, as I'll get into also, is also known to have deficiencies in DNA repair processing. And so in all of these, DNA damage accumulates in the nucleus. This damage then causes the pyrrolation process, and the process depletes the NAD pools, depletes functions of other proteins needing NAD for their function, and leads to mitochondrial dysfunction, not only in the form of mitophagy, but also other fusion fission, for example, have been connected with this as well. We think it's a nuclear process of DNA damage recognition and not a mitochondrial damage recognition, and I could discuss that later if anyone's interested. So then we came across ways to boost the NAD in the cell, and the main interventions that we've been studying are NAD supplementation in the form of nicotinamide riboside, or NMN, and these stimulate mitochondrial mitophagy function and other mitochondrial functions, and in fact also stimulate DNA repair. We were not the first to show this effect on DNA repair, but have done a number of studies that confirms that there is a direct role in DNA repair. So by using these interventions in model systems and in humans, we hope to restore mitochondrial function and boost DNA repair. And one example that we work with at the preclinical level, or hoping to work with on the clinical level as well, is Copenhagen syndrome, which is this very rare disorder of premature aging and severe neurodegeneration, and all of these features shown here, cachectic dwarfism. And importantly, in these patients, there has been, in 2 major clinical studies, hearing loss is one of the key features in this condition. So the patients have pretty severe hearing loss. Importantly, or interestingly, There are two clinical complementation groups, CSA and CSB. And the CSB and A protein functions differ quite significantly in their operation, but the underlying clinical features are similar for CSA and CSB patients. And CSA and CSB operate in transcription and what we call transcription-coupled DNA repair, and now also in mitochondrial functions. So I just here we did some work with finding that there is a mitochondrial deficiency in CS patient material and mice and CS depleted cells and the nematodes that we've used widely for model system, very useful model system. And then when we complement it with NAD, we could restore many of these functions. So at the cellular level, we saw, and in certain mouse models, we saw a beneficial effect of NAD complementation with nicotinamide riboside or NMN. And here's a mouse study where we actually normalized the mitochondrial features after treating the mice with NR, nicotinamide riboside, in the water. So the mice have some of the features of the human, but many others they don't have. But one that they do have is actually a hearing loss, a substantial hearing loss. So we did ABR, hearing loss studies in mice. We did the two complementation groups, CSA and CSB. And we took the mice at 5 weeks, and then they treated them just very briefly, 10 days, with nicotinamide riboside in the drinking water. And this shows the hearing deficiency at 5 weeks of age in CSB mice in this case, but we have similar results for the two complementation groups, CSA and CSB. And then when you continue the next 10 days, you see that NR avoids or NR prevents the hearing loss that you see in the CSB mice here without treatment. So complementation really stops the continuing hearing loss in this mouse model very efficiently, even after 10 days. And we did that in the CSA and CSB mice. We did a number of studies that are published a few years ago in a paper characterizing this deficiency in the cochlea. We found that there's a deficiency of ribbon synapse protein function. We found synaptic deficiencies, and we characterized further the inner and outer hair cells in that study. But we have, as we heard about today, the age-related hearing loss is of course such an important problem in our society. So we wanted to do an age-related hearing loss study with NR or NAD supplementation intervention. And the type of hearing loss in age-related hearing loss is sensorineural, and that's also what is seen in Copenhagen syndrome. So we could go on to use some other mouse models and do an age-related hearing loss study. And this paper was published last year, so I'll just highlight a few things from it. We found when we looked at NAD levels in the cochlea in the mice we did in CSA and CSB mice, we found that there was a decline with aging, which is what we would expect there would be. And then when they were treated with this NR, there was an increase here. So we prevented some of the hearing loss, loss of NAD in these mice. And this is just measured in different ways. And so in this study, we used 3 different mouse models. It's well known that the cadherin mutation in C57 black can be an issue. So we looked at different other models. We have an MT-KEMA model, which is a mitochondrial marker that we used for other purposes, and CBA mice in different studies. And here is what we found when we treated them long-term with NR. We saw a significant difference and improvement with age-related hearing loss decline in the ones that were treated with NR. And this was We also looked at the amplitudes of the brain waves, and in brainwave 1, you see a significant increases in amplitude after NR treatment, and this might then have a relation to synaptic function. And this is something we have looked at in a number of ways in these studies. For example, here in KEC pathways, synaptic pathways were significantly upregulated after NR treatment and were downregulated in the mice in general. We then also looked at imaging in various ways. We looked at the ribbon counts. We didn't see a lot in the base here, just about flat. When we looked at orphan ribbon counts, we saw a tremendous decline with the use of NR. And I'll get back to that a little further. So in synaptic related activities, and in fact, these ribbon synapse proteins have an NAD pocket, and they're also found in the retina and the eye, so maybe they're particularly receptive to NAD supplementation. In looking further at KEC pathways, we again found that synaptic related pathways were deficient and particularly improved after NAD supplementation. We looked at a number of proteins, pathways with nanostring approaches and with Westerns and functional approaches. We did not see dramatic upregulation of mitochondrial pathways, which we had expected, but we saw some upregulation with aging of oxidative-related processing proteins, and that they were less upregulated after NAD supplementation. So these are some of the conclusions from the work, and it was, as I mentioned, already published if you are interested. So I'd like to move over to some work that we have done with Alzheimer's disease. And that is, of course, a very important societal problem as well. And I think that it's particularly interesting that mitochondrial dysfunction seems to play an important role early on in Alzheimer's disease. And there seems to be some wide consensus about that. We have worked on DNA damage-related pathways, mitochondrial dysfunction in mouse models and nematodes and cellular systems. And mitochondrial dysfunction probably plays into the neuroinflammation process together with the DNA damage accumulation. And the neuroinflammation process is of course, very important in Alzheimer's disease. There has been many studies trying to connect DNA damage formation, DNA damage repair to conditions of Alzheimer's disease. But it's not that simple that in Alzheimer's disease, there's just less DNA repair. It's a complicated picture, but I would say that there's widespread agreement that there's a dysregulation of DNA repair. Sometimes something is upregulated, sometimes more commonly it's downregulated, but in any case, upregulation, downregulation of a critical step in the DNA repair process will end up limiting the DNA repair. And there has been some connections between Alzheimer's to mild-cognitive impairment to control of relatively more damage here than there than here. So we have worked with this for a while, and amongst the DNA repair pathways of particular interest is basic scission repair, the pathway that repairs oxidative damage in DNA, because oxidative damage is probably one of the most important to occur in the brain in relation to Alzheimer's. It's a simple process of a few essential steps. Some of these enzymes, APN nuclease and polymerase beta, are essential, and mouse models without these will not survive. So we took human tissue from postmortem brain from humans and analyzed DNA repair in general and in mitochondrial tissue also, and found that in these humans, the gap filling stage, the process here that involves the polymerase step of DNA repair, was deficient in the humans. And so that led to us developing a mouse that took several years to develop. It's a standard 3xTG Alzheimer's mouse in which we deplete that critical enzyme for polymerase and DNA repair called polymerase beta, an essential enzyme. And because it's essential, we can't complete it 100%. So it's a heterozygote 50% depletion, and this mouse has less DNA repair. This mouse has several features that differs from the 3xTG and makes it more humanized. It has neuronal cell death, decreased hippocampal volume, and all these features that are shown here. And interesting loss of olfaction, increased neuroinflammation. And so it's a humanized AD model, if you will, of a mouse. And we did a lot of work with this mouse where we looked at intervention with nicotinamide riboside and found that we saw significant improvements of the Alzheimer's features. We saw less damage, less tau phosphorylation, less neuroinflammation when we treated with this. So we've done a lot of work with this mouse model looking at AD features in our intervention. So now I'll just finish with this paper that was just very recently published, in which we looked at hearing loss in this Alzheimer's mouse model and connected it with some increased DNA damage, going back to the idea that DNA damage may be important in relation to hearing. And I'll mention that Laureen Sullivan has a poster, just showed it today with some beautiful imaging from this work. So I won't talk particularly about that. But in the mice, we then have a wild type. We have one that's just Pol beta deficient, doesn't have a very dramatic phenotype alone, and it's 50% deficient. Then we have the 3XT Pol beta, and then we have the 3XT mouse, and then we have the 3XT Pol beta mouse. So we were comparing really 4 mouse models. And what we found that was interesting to us was that we saw a dramatic hearing loss just at 4 weeks of age in these mice, much earlier than when we see cognitive impairment, smell loss, plaques, and all the features of Alzheimer's. Much earlier did we see this dramatic hearing loss deficiency. And it, in fact, is related to that the AD mouse has this here, but the Pol beta AD has even more of a hearing loss, connecting that if you don't have, or suggesting a possible connection between lack of DNA repair capacity and more hearing loss. We did some exposure of these. This paper is just out and you can read it if you have time. We found a more significant deficiency in male and female, which is a little surprising because in humans, well, this is AD is more common in females, but actually hearing loss, of course, is more common in men. But why the AD females exactly have more hearing loss is not entirely clear to us. We also looked at brain waves here and found some interesting deficiencies. And then we did a comparison, which was something idea here was, could we look at different areas in the brain? So we have the cochlea, and then we have an adjacent cortical area. The question would be, is the cochlea maybe a sink for DNA damage? If you get more damage formation there than other places, that could lead to age-related hearing loss and disease-related hearing loss. And in the field of DNA repair, it's well known that there can be differences in damage around different cells and genome, but it's very poorly really understood how that works. And so we just began a characterization of this, and the signal of DNA damage response correlation was dramatically increased in the cochlea compared to the cortex. And we also looked at nanostrain protein transcription patterns, and we found here, you can see it's very dramatic, that in the cochlea, there's much more of a DNA damage response than in the adjacent auditory cortex. And this shows correlation patterns were increased, and we did some other DNA repair measures. We found that sirtuin expression was increased, and I guess I will skip some of the detailed data here. We found PGAM was particularly lower, more or less expressed in the cochlea than in the cortex. We haven't really sorted out why that was. We found that NAD depletion was more dramatic in the cortex than in the cochlea than in the adjacent cortex. And so in conclusion of this work, we see this very early hearing loss in the Alzheimer's mice. That may be an important connection of others that have been also made with hearing and AD. And then we see this increased damage signaling in the cochlea region, very close by to another region in the cortex. And we see sirtuin activities. These are very important for mitochondrial function being deficient, suggesting a possible connection and with DNA damage happening, particularly in the cortex and in the cochlea. So I'll just, this is more general discussion of Alzheimer's and DNA damage. And I'll finish here and mention many of the people involved with this work here. We have a group in Copenhagen. We have members of my recent lab at NIH. And this was particularly Jay Hoon Park's work that I mentioned with the damage. Mustafa Okur did the age-related damage and the damage within the Cockayne syndrome. And I acknowledge this, these associations here and our other many collaborators here. I also want to acknowledge Uri Manor and his team in this last paper here. They did a very important contribution. Thank you so much.
For a couple of questions.
Yeah, actually, that you check any like UPR signaling pathways. So whether does like affect the gender translation, like
In the mitochondrial
yeah, mitochondrial. There's a nuclear pathway, related to the mitochondria, like ATF4, AT5. Yeah, pathway
in
UPR.
connection with this general signaling, we have looked at that as well, and we have looked at the signal as well. There's some deficiency in the UPRs also.
What effect does oxygen chambers have on DNA damage? As far as the healing process, that's what I was going.
So you mean we get a beneficial effect of oxygen chambers, but we also get increased DNA damage, and how does that counter that kind of? So interestingly, I mean, it's a very good question because oxidative stress to the genome can be tolerated to a fairly high degree. For example, these paraquat birds, they fly at very high oxygen conditions, and they have 30-fold levels of 8-oxo-G oxidative damage in the DNA, and this seemed to function pretty well. I think the threshold for when you really cause a problem with damage to DNA on the short term is fairly high. Maybe not so much in the very long term.
But it's a negative effect then.
It must be a negative effect. I mean, you don't want to get more damage into the DNA, but how much more damage can you tolerate? But I don't think there's any beneficial effects of that. There are beneficial other effects of high pressure oxygen. Let's say, yeah.
Enzyme deficiency is related with Alzheimer's disease. And just I am thinking, because monoamine enzyme system is FAD dependent, I think NAD supplement is not helpful.
To stimulate oxidative, what kind of oxidative function are you thinking about? Some kind of oxidation process? Yeah. I have not seen that we see that with, I mean, we look for some of those things. We look for oxidative damage as a marker and oxidative stress, mitochondrial ROS formation mitochondrial rust formation is not increased with NMN or NR, for example. But you're right, maybe there are some. It's such a wide system that could
Yes.
something.
Questions? Right, a round of applause.
Thank you.